All right, thank you everyone for being here. Welcome to this EvoFunPath seminar, and thank you to our speaker, Eugene Fletcher, for agreeing on coming and telling you about his work. Um, Dr. Fletcher is an assistant professor at the Department of Biology at Carleton University, where we did our retreat last summer. And prior to joining Carleton in 2023, he was in research and development at Escarpment Laboratories, a yeast company based in Guelph in Ontario. And with them, he's spearheaded an active research program to develop yeast and yeast strains to craft beers in Canada and abroad. And some of these strains now are used for brewing beers. Eugene did his PhD in cell biology at the University of Edinburgh in Scotland. And in 2014, he did a postdoc with Jens Nielsen in Sweden, sorry, in Denmark. And then he also worked at University of Ottawa. For those who know yeast synthetic biology and metabolism, Jens Nielsen is a leader in the field. His research program now at Carleton aims at engineering yeast with using synthetic biology and constructing synthetic metabolic pathways to upcycle plastic and dairy waste into valuable chemicals, biochemicals, and also is examining how the resulting cellular stresses that shape fungal adaptation, trade-offs, and tolerance. In addition to his experience in academia and industry, Dr. Fletcher has developed products that are under US patents, and is also a passionate scientist trying to move ideas from the bench to the market. So I think it's a great example for us. Also, with your experience in industry, I'm sure you'll have some good advice for our fellows in the discussions after the seminar. So thank you again for being here, and thanks everyone for joining.
Right. Thanks a lot, Christian and Marie-Hélène, for this invitation. I'm very happy to be here today to share my work with the group. So, yeah, as you mentioned, I'm at Carleton University, and I moved here just about 2 years ago from working in industry. So my group is still very young, very small. We have this nice new lab space because we've just been there for 2 years, and we hope we keep it clean as it is looking right now for years to come. Yeah, so in my lab, we focus a lot on molecular microbiology, where we try to understand different biological processes that happen within the cells, especially for yeast cells, and with the intention that we can use that for some application in industry or in medicine or for some other interesting use outside the lab. So as you can guess, yeast is our favorite member of the fungus family. I know most of you work on fungi or fungus or fungi. Yeah, but yeast is our favorite member, and it's been used for several years. We've evolved. We have co-evolved with yeast for several years, so much that they've actually impacted the food that we eat and the kind of things that we drink. So right now, the most widely used yeast strain that we have in our lab is Saccharomyces, but we're also looking at some non-conventional yeast strains that haven't been too well characterized for different applications. So if you look at the yeast cell under a microscope, it looks like this round circle I've shown on the slide. And for some people, when they see this round circle or this round cell, what they think of is it takes up sugar and then produces alcohol or organic acids at the end. But there's a lot of magic that happens in that white space. So if you look deeper into the cell, what you'd see is something like this, where there's a lot of interconnected biochemical pathways that interact in a very complex way. So much that if you understand how these pathways work or how they are connected, you can actually engineer the cells with new information that can make them produce things that they do not produce naturally, like drugs. So I have a very quick question to everyone here. If you can go on Slido, um, type in this code, or you can also scan the barcode. Let me know what you think. If fungi were to vanish tomorrow, which of these do you think would collapse first? I want to know what you think. So I'll give you a minute and then I'll take your answers. Yeah, I see a lot of giggles in the in the room, and looks like most of you are done. Let me see what the streets are saying. See. All right, okay. uh, yeah, so it looks like most of you are saying that, yeah, the forest ecosystems, of course. And then there's a beer and wine, the global carbon cycle as well. But yeah, of course, the forest ecosystems. All right. Yeah, we'll class first. Interesting. All right. Um, right. So those are all very good answers. Those are all true. But in my lab, we are trying to find a new use or a new application for yeast, where we are trying to explore the possibility of engineering yeast cells or like fungal cells to convert plastic waste into useful biochemicals. So normally or naturally, yeast cannot do this. So what we are trying to do is to engineer a synthetic pathway, stick it into the cell, and then train it to be able to recognize plastic compounds as a carbon source, and then ferment this or convert this into ethanol. So of course, there are a lot of things we need to tackle. There are a lot of questions that we have. For instance, like how would yeast rewire its metabolism to express this foreign synthetic pathway? And again, how is it going to deal with resource allocation? Now, you are telling it to produce new enzymes it has never produced before. How are you going to? How is the cell going to allocate resources to express these enzymes and make it active? Also, what are the intracellular stresses will the cell go through? So these are some questions we have. We are continually asking the lab, and we are actually working towards these. So just in case you haven't heard it before, yeah, let me be the first to tell you that we have a plastic pollution problem. So plastics are everywhere. It's a big problem now. Different governments are trying to find different ways to solve it. In Canada, there was some talk a couple of years ago to ban single-use plastics, which didn't go too well because there are some plastics that you cannot do without. So now we need to find good ways to deal with the plastics. Some people have talked about recycling, but then when you recycle plastics, some of it gets retained in the environment and it's not really solving the problem. So myself, I am originally from Ghana and on the west coast of Africa. So I do go back home every year to go see family, and usually when I go back home, I take a little stroll along the coast because yeah, I'm from the coast. So take a little stroll along the beaches. So back in the day, if you go to the beaches, they look like very pristine, very nice, very clean. But recently, if you go to the beaches, you see a lot of plastics that have been washed onto the shore. And of course, the scientists within me went around looking at the labels in the plastics just to see where they're coming from. And interestingly, most of the labels were in French. And that means a lot because so Ghana is English speaking and every country around us is French speaking. So to see these labels that you can't recognize and also are in French suggests that they've been probably washed from the shores of neighboring countries onto the shores of Ghana. So all of this is to say that the plastic pollution problem is a global problem. It's like we are all in it together. So then we all need to work together to solve this problem. If you come home to Canada, most of our plastics, you might know, it's not recycled. So eventually they get into the environment, they break down to microplastics, and then end up in the food and water that we drink or consume. And actually, so just last week, I think on Friday, this paper came out in Nature Medicine. And I think this is the most comprehensive paper I've ever seen published yet on the effect of nanoplastics and microplastics on the human body. So they did this very comprehensive review where they showed the effects of these plastic compounds on different organs, like the lungs, the kidneys, the intestines, even the placenta, and other organs within the body. And all of this to show that there is a problem, and we are sort of might face some health challenges in future if you don't really check the pollution that you're putting out there in terms of plastics. So a few years ago, maybe 9 years ago, this article was published, and that brought a lot of excitement within the field because some scientists in Japan had isolated some bacteria from a landfill site. And this bacteria they found was able to consume plastics as a carbon source. So that brought about a lot of excitement, and then that led to a whole new research field of finding the enzymes that break down the plastics into the basic building blocks. So in my lab, we have been looking into that. But to understand how enzymes break down plastics, we need to understand the chemistry of plastics. So plastics are essentially long chain polymers. They are made up of small basic units or monomer units that are joined to form a long chain. Pretty much like how glucose units would form long chains of long chains to form like carbohydrates. You have the same thing in plastics where there are smaller units that come together to form these long chains. So if you have the right enzyme that can break these chains, you'll be able to break down the polymers or the plastics into the most basic building blocks. So for instance, for PET plastics, like the plastic bottles and cups that we have, with the right enzyme, you can break down these long chain polymers into these basic units called TPA or terephthalic acid and ethylene glycol. In my lab, one of my students, undergrad students, was very interested in looking at some of these enzymes that break down plastics. I sort of like move away from this research because everyone is doing it. It looks like very crowded at the moment. But this student was like very keen on learning about like how enzymes work and how to engineer enzymes. So he approached me and we came up with this project that actually worked out better than I thought it was going to work out. So most of the enzymes that we know break down plastics are from bacterial sources, or because we've only found or studied bacterial enzymes that break down plastics. So we found a paper that was published last year on a fungus, a mold, that can produce a cutinase enzyme. And in that paper, they show that it's able to break down PET plastics and some polyurethane plastics as well. So what we did was we tried to improve this enzyme by using directed evolution, where we just used an error-prone PCR to introduce random mutations into the enzyme. And then we did a big screen on agar plates containing a substrate called tributyrene. So tributyrene has the same chemical bonds that you find in PET plastics. So if your strain is able to produce an active cutinase enzyme that breaks this bond, you see a clear zone of inhibition around the spot where you drop the strain. So we did that and we measured the diameters of the zones of clearance, and then came up, found one mutant that was actually producing a more active version of the enzyme. We sequenced the gene that we sequenced the cutinase gene that we put in the strain, and then looked at sequences and saw two mutations in the functional domain of the enzyme. So I don't know if you can see my cursor, but it's around 178 amino acids, and then around 218 amino acids. So this enhanced cutinase had these two mutations that were in the functional domain of the enzyme. So we're still trying to characterize it, trying to understand how these mutations impact the structure of this fungal cutinase and why it's more active than the original cutinase. So just back to the enzyme story. So if some companies like Carbios in France, they are also using AI and machine learning models to improve some of the enzymes that break down PET plastics into the basic building blocks. And for them, the idea is that if you break down PET plastics into the basic blocks, you can repolymerize them into new plastics using these same basic building blocks, which is a fair way to go. But I have a different opinion because if you reconvert old plastics to new plastics, some of the plastics will belong along the chain and would end up in the environment. So what if you can convert plastics to something very different, but of a high value, but it's of high value nature? So for us, instead of breaking up plastics to these building blocks and then converting them into new plastics, in my lab, we're looking at ways by which we can use these plastic building blocks as a carbon source for yeast fermentation, where we can convert them into some interesting compounds. Right now, we're looking at converting them to bioethanol because yeast to produce bioethanol naturally. But once we perfect this pathway, we will look into producing some more interesting compounds like flavor compounds or fragrances or scents using fermentations where we convert plastic compounds into these interesting compounds. So basically, the scent in your shower gel could come from some waste plastic that someone dumped into the recycle bin maybe a week or two ago. So that's our idea, where we want to convert the plastic compounds into some more interesting and high value compounds. So Flor in my lab, she's a grad student looking at building a synthetic pathway in yeast, where we're looking at taking different enzymes from bacteria that have been confirmed to consume plastics. So we're picking these enzymes, building like a synthetic pathway, which you're going to stick into yeast so that yeast will be able to recognize plastic compounds as a carbon source and then convert these compounds into pyruvate. And then from pyruvate, we can can channel the carbons into ethanol or biofuels. So that is going, but there's so basically using synthetic biology tools, and my analogy I use here is that we combine the pathways or the genes in the pathways, pretty much like how the mobile phone companies would combine different parts into a functional mobile phone. So basically, you can train the cell to do something that it doesn't do naturally by combining these new parts. But of course, like every science project, there's a bottleneck. So yeast have evolved over time to produce alcohols from sugars. They have not really been exposed to plastics enough for them to adapt to the toxic nature of these plastics. Because these plastic compounds are very recent compounds that we have been producing. So we were challenged with the fact that TPA or terephthalic acid, which is one of the compounds from PET plastics that we are trying to convert, is actually very toxic to the yeast cell. It kills it. So that is a challenge because it's basically like we giving the cells like this poisonous food to eat and expecting it to produce something out of that. So we need to find ways by which we can make the cells more resistant to this poisonous or toxic carbon source that we give to the cells. So our attention really changed a little bit where we focus more on improving understanding why terephthalic acid is toxic or poisonous to yeast. And once we find that, we can engineer our strains to be more resistant, and then that will impact the yield of the product that we're trying to produce. Because if they are killed, if the yeast cells are killed by the compounds that they're supposed to consume, then of course the yield of the ethanol that we're trying to produce will be low. So we need to increase the resistance or tolerance to these toxic compounds. So that is one question we've been asking or trying to look into for the past year and a half, where we are trying to find out why terephthalic acid is toxic to yeast and what biological mechanisms modulate this compound's toxicity in yeast. So it's an acid, and there's a lot of published work on how acids impact yeast. There's also a lot of published work on how compounds with an aromatic structure, like this one here in TPA, impacts yeast. But there's nothing known on any compound like terephthalic acid or TPA that has both an aromatic compound and two acid structures on it. And yeah, that the impact of the combination of these structures on yeast. So yeah, we had to look into it. And just going off topic a little bit, I finally watched this movie last weekend. Highly recommend. And the storyline is pretty much like our story here, where the yeast cell is more or less like the hero, or it's going to be the hero of this story, and then the villain or the bad person is the terephthalic acid or this plastic compound you're trying to work with. So this whole story is that I'm going to tell now is about how the yeast cell is trying to fight the toxic effect of this compound and what tools or mechanisms it's going to use to fight this toxic effect of the compound. So we are trying to use this design, build, test, learn cycle where we like to engineer some genes or some pathways in the yeast cell to make it. So I'm getting some echoes.
I think it's fixed now. Maybe it was Richard's microphone.
Okay, thank you. Yeah, thanks much better now. Yeah. So we're trying to use this design, build, test, learn cycle to improve a yeast cell so that we can push the limit of resistance from this blue line to the red line. And to use this design, build, test, learn cycle, we first need to know what genes or what biological pathways to engineer. And then once we engineer that, we do a lot of testing to see if it's working or not, and then learn from that data to build or to further improve the strains. But we are at a spot where we don't know what pathways we should target because nothing's published on that. We don't know what genes we should target because there's no information on that. So then we have to find these genes or pathways ourselves. And to do that, we are using all the tools that we have in our hands. So we are using RNAseq to find out what genes are turned on or off when yeast cells are grown in the presence of TPA or this plastic compound called terephthalic acid. We also have a collection of these deletion mutant libraries, which contains over 4,000 yeast mutants. And each of these mutants has a non-essential gene deleted. So we're trying to screen all of these mutants on TPA to see which strains are hypersensitive or hypertolerant to the toxic effects of TPA. And of course, also using lab evolution, where we allow nature to do the hard work and tell us what genes or pathways to target in terms of improving resistance to terephthalic acid. So for our chemogenomic screen, we screened a library of over 4,000 yeast mutants. So this is how it works. So the library, which you see on this in this video, is this plate that a robot hand is going to pick. So each colony or each dot you see there is a yeast mutant. And each of these mutants has a non-essential gene deleted in it. So we pin that library onto two plates, a control plate that has nothing added, it's just YPD. And then we'll pin the library onto the selection plate that contains a toxic amount of terephthalic acid or TPA. Grow the plates for 3 days and then look at mutants that show hypersensitivity or hypertolerance. So if you look at the plates on the right, the selection plate, you can see that there's some patches with no growth. And that shows that those mutants at that spot are hypersensitive to the chemical. So we can trace back to see what genes they are lacking. And then that gives us an indication that those genes are very important for growth in the presence of TPA. So, I mean, you can use a robot to do this. If you don't have a robot in the lab, which I don't expect most labs to have, you can have a different kind of robots called undergrads. Give them this 96 handheld pin, and an afternoon, they can pin the collection from the stock plates or the libraries onto the selection plates using this handheld pin. And this also works very well. So we did that and then came up with this chemogenomic profile. So what you see on this slide is different nodes. So every node or cycle you see here represents a gene that, when deleted, makes the cell resistant, makes the cell hypersensitive to terephthalic acid. So we were able to classify the genes into different sort of functions, like amino acid biosynthesis, DNA damage. There are some genes that play a role in transcription regulation. And there are some other genes that we couldn't put into any of these classes, but then they also play different roles within the cell. So from this list, we did some Go term analysis where we tried to find the biological processes where these genes play a role in. And we saw that most of them, or a lot of them, were kinases, and some of them were also oxidoreductases. We also found that some of them played a role in DNA binding or DNA regulation. One interesting thing that came up from this list is that a good amount of the genes in our list were phosphoproteins, which means that they are either phosphorylated or they phosphorylate other proteins within the cell for whatever function. And that's the first indication that phosphorylation is probably very important in maintaining growth in the presence of the toxic concentration of terephthalic acid when it grows cells in that environment. So another thing that's also striking is if you look at the blue nodes where that represents the amino acid biosynthesis, you notice that 3 out of the 4 play a role in threonine biosynthesis. So that got us interested in looking deeper at that pathway to see why threonine biosynthesis is very important for growth in the presence of terephthalic acid. So we did a dot assay where we tried to validate the mutants that we found in the screen, and then also check out the other genes in the pathway that didn't come up in the screen to see if they also played a role in they also played a role in modulating TPA toxicity in cells. So we did a dot assay where we spotted these mutants on YPD agar plates containing no terephthalic acid, and then on YPD plates containing terephthalic acid. So you can see that on the plates that had the acid, our mutants that came up on the screen were hypersensitive. They showed no growth on the plate. And looking back at these genes or these enzymes, the pathway, so they catalyze some very key steps in the threonine pathway. So HOM3 catalyzes the rate limiting step in the pathway. So once the step is passed, the pathway will happen. And there's also HOM6 that leads to the formation of homoserine, and THR1 that also converts homoserine into threonine through to a couple of steps. So this was a good indication, a good confirmation that these enzymes are really needed or are vital for growth in the presence of TPA. But just hold on to this thought. I'll come back to it in a second. If you go back to look at the chemogenomic screen, you also notice that there are some genes that played a role in DNA binding or played a role in transcriptional regulation. So that's made us think that it would be very interesting to see how the cell sort of rewires its transcription to deal with the stress. So we did an RNAseq experiments by growing the cells in the presence of TPA, and then looking at all the genes that are turned on or off in the presence of this toxic compound to get an indication of like what pathways are really needed for survival under these conditions. So we ended up with several genes that were induced or downregulated in the presence of terephthalic acid. What was striking was that a lot of the glucose metabolic genes, like all the high affinity glucose transporters, were highly induced. And I'm talking like 7-fold induction. And then we also saw some of the phosphate metabolic genes also upregulated, like 4, 8, and 9. It's a phosphate transporter or transporter that is found in the membrane. Was also very highly induced. But interestingly, some of the phosphatases were also like very highly downregulated, which was interesting. And also sort of gave us a hint that we should look at phosphate metabolism. Because from the chemogenomic screen, we saw that most of the genes that came up in our list were all phosphoproteins. And we also seen some role of phosphate metabolism in our RNA-seq as well. So yeah, give us an indication to look into that. And as you'd expect, a lot of the stress response genes were also highly upregulated, like the oxidative stress response, the heat shock response, like all these general stress responses were also very highly upregulated. And most of these genes played a role as oxidoreductases or peroxidases, which also confirms the role of oxidative stress, a chance that there's some oxidative stress going on within the cell when they are exposed to terephthalic acid. So because a lot of the glycolysis genes were upregulated, we looked at we tried to map the glycolysis pathway or the glucose metabolism pathway to see which of the genes or the enzymes in that pathway were upregulated in our screen and try to make some sense out of it. So the HXT5 and 7, which are the high affinity glucose transporters, were highly induced or highly upregulated in our RNAseq data. And also the hexokinases, the rate limiting step in the pathway was also highly upregulated in our data. We're also seeing all the cholesterol metabolic genes or enzymes or genes rather were highly upregulated. And then interestingly, the genes that code for enzymes that converts or makes the cell consume cholesterol were also induced in our data. So the genes that are colored purple are also the genes that are phosphorylated during glycolysis. And you can see that nearly all of them that were upregulated in our screen are also naturally phosphorylated and drive flux through the pathway through glycolysis. So in glycolysis, the main driving factor that moves flux down is the role of phosphorylation of the key metabolites in the pathway. So we're wondering why glycolysis was highly upregulated or induced, but then the growth of the cells, but then there was like a growth defect. So we measured the growth rates again in the presence of terephthalic acid and YPD, the control, and saw that indeed there was like a reduction in growth, of course, when you grow the cells in TPA. But we're wondering what was going on. So we looked at the glucose uptake rate to see how well the cells uptake glucose, because you're seeing all these genes highly upregulated. That translates into how well they consume glucose. So we looked at that and we were shocked to see that cells grown in the presence of terephthalic acid, shown here in orange, actually have a much lower glucose uptake rate. They have like over 50% reduced uptake of glucose compared to cells grown in YPD without the compound. Which was interesting because you see all these genes upregulated. So then why are they struggling to consume glucose? So that sort of gave us an indication that probably terephthalic acid is inhibiting some mechanism required for glucose uptake, which we do not know yet. So we're still trying to look into that. But as I mentioned, glycolysis, flux through glycolysis is really controlled by phosphorylation. And we're already seeing a lot of these phosphate metabolism genes in our chemogenomic screen and also in our RNA-seq data. So we wanted to look more into why or how the cells rewire its phosphate metabolism to make it able to deal with the stress of growing in terephthalic acid. So under phosphate limited conditions, this is like from several published work, when phosphate is limited in the cell, in the media, all the genes, the full regulin or the phosphate genes are all highly upregulated or induced. So the transporter is induced, the co-transporter, the NAL1, is also induced under phosphate limiting conditions. And also the phosphatases are all induced because the cell tries to maximize its gain of phosphate to deal with all the metabolic processes that needs phosphate. But in our work, we're thinking that probably there's also some phosphate limitation, but we saw something that was off. So the phosphate transporter was actually induced, like you see under phosphate limited conditions. But then all the phosphatases that are usually upregulated during phosphate limitation are downregulated in our data. So that tells us that the cell probably prioritizes getting phosphate from the environment into the cell and limit breaking down the metabolites that have phosphate in them so that they can still function, because they probably need those phosphorylated metabolites to deal with the stress, probably by, and some of them produce like ATP, and this also regulates other pathways or control other pathways. So those pathways are maintained, they are sort of like preserved, their phosphates on there are preserved, and the cell would prioritize gaining or bringing phosphate from the environment into, or from the media into the cell by upregulating its 489 gene. So pretty much that there's some sort of like rewiring of this phosphate metabolism to make the cells able to increase the phosphates that it needs to regulate different pathways required to deal with TPA or terephthalic acid stress within the cell. So again, we measured the phosphate uptake rate, and so that the cells that were exposed to terephthalic acid actually had a much higher phosphate uptake compared to the control cells without the acid, which confirms the role of this phosphate transporter, which has been highly induced in the cell, and saying that it's actually bringing in a lot of phosphate at a much faster rate within the cell to regulate different pathways that require phosphate to function and to preserve the cell in the presence of terephthalic acid. So back to this threonine story, if you recall, 3 of the key enzymes in the pathway, when deleted, makes the cells very sensitive to terephthalic acid. Interestingly, 2 of them, home3 and THR1, are also phosphoproteins. They are phosphorylated to function, so they need phosphate to function. And also, when they're deleted, of course, the cells would become very sensitive to the compound. But what was striking for us is that, so under normal conditions, these mutants grow very well. They grow similar to the wild type. But when they are grown in the presence of terephthalic acid, then there's a hypersensitivity. And that was an indication that there is probably an accumulation of some of these metabolites in the pathway when the genes are deleted. That's what causes the toxicity. But the question is that if these metabolites are toxic, why are they toxic only in the presence of TPA and not under normal conditions? So the simple answer is that it's probably because in the presence of TPA, flux through the threonine pathway is much faster. So that pathway is much active during growth in TPA. That leads to a higher accumulation of the toxic metabolites that the cells struggle to live under. And actually, there's been a lot of some published work in the past that show that for some stresses like salt or even high pH, when these threonine mutants are grown, because of the high flux in the threonine pathway, there's an accumulation of homoserine, which is the main metabolite that causes the toxicity. And it's toxic because it has a similar structure to threonine and serine. So it acts as a threonine analog, which can fit itself within a protein chain that requires threonine or serine. So this study that I've put up there, this 2010 study, showed that when you have some of these threonine mutants, they accumulate homoserine, and homoserine is toxic because they insert the cells or they replace threonine in protein chains, and that leads to a loss of function of protein, and the protein also gets degraded. And in our case, like or TPA, where phosphorylation is very important for the cell to survive under this toxic compound, imagine replacing threonine, which is the residue of protein that's phosphorylated. Imagine replacing this residue with homoserine that is not phosphorylated under these conditions. You deprive the cells of that phosphorylation that it needs to regulate all the pathways like glycolysis or ATP synthesis that the cell needs to survive under this toxic environment. So homoserine replaces threonine, it gets the protein degraded, and also the protein loses its function. So if homoserine is a problem, why is HOM6 also sensitive to TPA? Because this mutant does not produce homoserine. So there's a paper that also showed that GCN4 is one of the regulators that regulates HOME3, THR1, and THR4. So in the presence of this protein, these enzymes are upregulated the fueling pathway functions. But in mutants that lack HM6, there's an accumulation of aspartate semialdehyde. And this metabolite inhibits GCN4. So when it inhibits GCN4, the enzymes further downstream, like THR1, are not able to function to they're able to function, so then homoserine builds up, and then you end up with the same problem where it becomes toxic because it replaces threonine in proteins and leads to protein degradation. So that is why HM6, even though it doesn't produce homoserine, it's hypersensitive because of this intermediate that it accumulates. And this intermediate, the aspartate semialdehyde, inhibits GCN4, and that's also when that's inhibited, all the enzymes downstream like THR1 is not able to function. So you have homoserine building up or accumulating within the cell, and that leads to toxicity. And that is what is going on with our strains that are growing in the presence of terephthalic acid, this TPA compound. So our model, or what we think is happening, so with these tools that we use, what we think is happening here is that in the presence of terephthalic acid, the cells are trying to produce ATP to fight the stress. But then they're challenged because the TPA inhibits glucose uptake. So then they have to use other resources to they have to like over-induce the glycolytic pathway to compensate for this reduced glucose uptake and glycolysis in order to produce enough ATP to deal with the energy, to meet the energy demands of the cell. And also key metabolic pathways like threonine biosynthesis, which are also phosphorylated, also require ATP. So then the cell would have to improve its ATP, its phosphate uptake to be able to get these other pathways running. You're also seeing an upregulation of catalysis, which deals with the oxidative, which deals with the buildup of ROS within the cell. And yeah, catalysis also requires iron to work to function. And we're seeing an induction in iron transporters in that data as well, which aligns with what we're seeing that the cell is going through oxidative stress and needs to have an active catalyst that requires ferrous iron to function to deal with the stress. So all of these is going on, but then the most important thing is being able to supply the cell with enough phosphate to regulate all the pathways that the cell needs to fight the stress. So since this is a fungal group, I was just looking at other other applications of the threonine pathway that could be of relevance to you. So we've seen that when some of these genes or enzymes in the threonine pathway are deleted, the cells become sensitive to the drug or to a compound, and could be a good target for an antifungal. And interestingly, in humans, we don't have the threonine biosynthesis pathway. So then this means that targeting this pathway in pathogenic fungi could be a good target because then there's a low chance of toxicity in the human host. So what would be an interesting thing to do is to find or to screen for a drug that increases flux through the threonine pathway so that there's an accumulation of different metabolites in the pathway when some of these genes in the pathway are deleted. So imagine you have a drug that increases flux and also inhibits, say, HM6 or 3. You'd have an accumulation of some of these metabolites that would kill the drugs, that would kill the pathogenic fungus or fungi. And in fact, some of these drugs have already been developed that target certain enzymes in the threonine pathway. So HS9, I think, targets HOM6 in the threonine pathway and has been shown to be very potent in killing pathogenic fungi. What hasn't been done yet is drugs that target THR1. And that'll be an interesting target because when this enzyme is deleted or inhibited, then you have the accumulation of homoserine, which is toxic and kills the cells, making it a very potent target for drug design. If you're looking for new targets for an antifungal, pathogen. If you're looking for new antifungal targets for a fungal pathogen, yeah. So also on the side, we're also using lab evolution to find out what genes or mutations would be very important in increasing resistance or tolerance to terephthalic acid in yeast, and basically allowing nature to do all the hard work to tell us what targets to look at. So right now we have just recently finished this lab evolution. It took us 65 days, and we've isolated three mutants you can see on the right that have shown to be very resistant to terephthalic acid. So the red graph or the red line is the wild type, the original strain that we used, and then the other colors represent the isolates we got from the lab evolved cultures. So we've just sent them for sequencing. We are still analyzing the data. We're seeing some mutations in some of the key transporters in yeast, suggesting that the cell is trying to mutate these transporters to limit the influx of terephthalic acid into the cell. And there is also one transcriptional factor, transcriptional regulation gene that we saw. I can't remember what name it. I can't remember the name of that protein. So this gene had a premature stop codon, which is a sign that the cell probably tries to block the transcription of several other genes by um, inactivating that transcription regulation protein. So we're still looking into this, and putting all this together will give us a very good and comprehensive idea of what happens within the cell when the cells are exposed to terephthalic acid, which you get from PET plastics. And with this, you can build a platform strain that's very resistant to the compound and use this platform strain to convert terephthalic acid into interesting compounds like fragrances or biofuels through fermentation. So I'm just going to end here and recap what I just presented, that terephthalic acid induces glucose starvation, and the cell tries to compensate for this by overexpressing all the genes involved in glycolysis and in glucose transport. And then we also see an upregulation in phosphate uptake. And you can also see the cells actually increasing its phosphate uptake in the presence of TPA to meet the energy demands of the cell, and also to make enough phosphate available for all the phosphoproteins that regulate other metabolic pathways within the cell that is required for growth in the presence of terephthalic acid. And you're also seeing that threonine biosynthesis is very important, or it's a very essential pathway in yeast, and should not be tampered with if you want to have a strain that can live in the presence of terephthalic acid, especially in the case where you're trying to modify different genes or bring a foreign pathway to make the cells convert terephthalic acid into new compounds. So you should make sure you preserve the threonine pathway and not tamper with it, because it's very essential, and the cell needs that for survival in the presence of the compound. So just going to end here. Thank all the people in my lab who've done this work, and all the funding we got from the ENSERC, from Carlton, from Global Affairs Canada to support this work. And of course, thank you all for spending your Tuesday morning with me to listen to the work that we've been doing. Yeah, I'll just stop here for some questions. You can also reach me on any of these platforms. Again, I still refuse to call Twitter X. I call it Twitter anyways. You can reach me there if you have any questions. And yeah, I'll stop here to take some questions. So yeah, merci beaucoup, and thanks for your time.
Thank you, Dr. Fletcher. Any questions?
I have a question, actually. I'm wondering, do you know how TPA gets into the cell? And does that have anything to do with phosphate?
Very good question. I get that question all the time. So we don't know because there's no published work on that. I get this talk in the conference last year, and that's like one question several people asked. So we don't know. We don't know yet. But what we think is it probably uses some of the organic acid transporters to get into the cell. So we saw that PMR1 has some mutations in a lab evolved mutant. There was also some mutations in Gen 1. So these are organic acid transporters that have been shown to be also to be broad spectrum. So they allow multiple or different kinds of organic acids into the cell. So we're thinking they're probably getting into some of these non-specific organic acid transporters. But yeah, we don't know for sure.
Cool. Thanks.
Yeah,
Hi,
thanks.
Eugene. Hi, Eugene. I enjoyed your talk, and nice to meet you.
Nice
So
to meet you, Richard.
I was wondering, so these decay fungi, these white rot decay fungi also break down plastics. And when I teach my forest pathology course, I mentioned that, but I never kind of looked into it. But are they also inhibited by the monomers like the yeast were, and is that like, are they sort of better adapted to this task than the yeast would be?
Mm-hmm. Yeah, that's a very good question too. So these white rot fungi break down plastics by producing enzymes that will break it down. But I'm not too sure how toxic these monomers are to the fungus. And also in the wild, there's also a very complex system out there because they break down the plastics into these monomer compounds. And you also have some bacteria that consume these monomer compounds also in the same environment. So there's a good chance that there's this interaction between different organisms where the white rot fungi produce the monomer compounds, which are used up or consumed by other bacteria in the same environment. So it limits the concentration of the compound in the environment, and probably the cells are able to live there without getting poisoned by it. But that's a very good question. I'm not too sure how toxic it is to them, but I'm pretty sure there are some other microbes that consume those compounds while they're being produced, and that reduces the concentration and then the toxicity of the compound.
Mm-hmm. Yeah. Thank you.
I have
Thanks, Richard.
a really small question concerning the slide showing growth of Ebb survey, comparing TPA with EBD. I noticed you used a concentration of 0.3 mole TPA, which is quite a high concentration, if I may say. My only question was the TPA the only carbon source in that condition, or was it added alongside glycose and UPD? Or in other words, is yeast truly metabolizing TPA or mainly tolerating it?
Mm-hmm. That's a great question. So, yeast cannot metabolize TPA naturally. So in that experiment, we also added glucose as a carbon source, and then we only tested the toxic concentration of TPA for toxicity or resistance. So those are glucose as a carbon source, and we're just looking at the toxicity of the compound. We did not expect yeast to metabolize it because they do not have the genes or the enzymes or the pathway to metabolize TPA. Right, Matthew?
Eugene, thanks for your talk. I wondered if you would there be a benefit to trying to increase sort of the sink strength for threonine? Like, could you make a polymer of threonine or use it up faster, and would that help avoid this toxicity?
Mm.
You know, could you add a new function to the yeast that would create a stronger sink for threonine, and would that help?
Mm-hmm. For sure. Yeah, that's an interesting one as well. I have looked into that. I've seen people report that if you introduce some specific mutations in HOM3, the rate limiting step, that drives the carbon flux much faster within the pathway. Because so what happens is that threonine sort of like inhibits that enzyme by feedback inhibition. So if you introduce some mutations in there, it can no longer bind to it, can no longer inhibit it. So it keeps the pathway like literally like open all the time and mostly faster. But that's true. To be interesting to create this like threonine sink to see if that would further increase resistance to TPA toxicity. Yeah, that's a good point. I'll be interested in trying that.
I'm sorry, there's a question in the chat from Dr. Subramanian. It's related to Dr. Amelin question. Have you looked at toxicity with other fungi, phytopathogens?
No, we haven't looked at that yet, but now that you mentioned like a lot of people are also showing a lot of interest in that, so that'd be an interesting, interesting mechanism to look at because, like I said, a lot of these compounds have like overlapping mechanisms, even to other drugs or compounds. So that would be very interesting to see if there's some overlapping mechanisms that's shared with other antifungal drugs or compounds. But we've not looked into that, but yeah, for sure that'd be very interesting.
There's another question. Oh,
Yeah, no, go ahead.
okay. So Mathieu Gigari is asking, saying thank you for your presentation. And then I have a question about TPA transport from outside to inside the cells. Do we know which transporters are used?
No, we do not. There's no information on that. But we're thinking that it probably uses some of these like broad spectrum or non-specific organic acid transporters to get into the cell. So, so mentioned, we are seeing some, from our lab evolved cultures, we are seeing some mutations in some transporters like JEN1 and PMR1, which are some of these broad spectrum organic acid transporters. So we're thinking it probably uses some of these to get into the cell. But yeah, we don't know how it gets into the cell. Yeah, good point.
Hi. Thank you so much for that. That was very cool.
Thanks.
I had a question regarding on like how you choose which fungus or which species you're gonna use as a model. Because if you have similarities between them, but you also, if you are using what's intrinsic to them and they have differences between them, and then synthetically changing that, how do you like what kind of characteristics, or I don't know, what are the things that you're looking for that will define what is a good model, if that makes sense?
Mm-hmm. Yeah. So so we're using Saccharomyces because that's a model model fungus, and it's been very well studied, very well characterized. We understand its genetics very well. It's also single cell, so it's easy to study new changes in that. So for us, that's the main reason why we're using Saccharomyces, because it's like a very simple eukaryotic cell to study biological mechanisms to different compounds or toxic compounds like terephthalic acid. And also, we're also using Saccharomyces because we would like to show as proof of concept that you can convert plastic waste into something, into bioethanol. And Saccharomyces can do that, can produce bioethanol very well. What is missing is the first part that's consuming the plastic waste. So once we get that pathway assembled and then connected to ethanol production, that'll be a good proof of concept that you can convert plastic waste into bioethanol. So to your question, we're using Saccharomyces because it's an easy, it's a simple organism to study, and also because it can produce ethanol very easily. But it'd be interesting to try other fungi that produce interesting antimicrobial compounds, and then try to see if you can link PET consumption to the antimicrobial compound production. Because we already know that some, Richard mentioned, that some fungi produce enzymes that break down plastic. So if you can link that into antimicrobial production, that'd be fantastic. Yeah.
Okay, thank you for your questions. Maybe for the sake of time, we're gonna stop here. Thanks again, Eugene, for your great talk. That was super interesting. And all the fellows are invited to stay and chat with Eugene. I think his background is quite different from standard academics that we've talked to. So it's a great occasion to know about move from the private industry to academia or vice versa. So thank you again for your talk. It was great.
All right, thanks very much. Really appreciate you having me here. Thank you very much.
Have a good day.
Thanks.
Thank you, everyone. Dr. Fletcher, if you want to take a sip of water or something, we can just come back in like 2-3 minutes, and Penelope will manage the Q&A with the fellows. I'll stop.
All right, thank you very much.
Well.
Okay.
Thanks. See you in a couple of minutes.